Apoptosis is a type of programmed cell death which involves a genetically determined elimination of cells. Apoptosis also occurs as a defense mechanism when cells are damaged by disease or noxious agents. There are a few markers that confirm the induction of apoptosis, including cell shrinkage, condensation of the nucleus, and membrane blebbing. The cells are smaller in size, the cytoplasm becomes dense, and the organelles are more tightly together. Since the plasma membrane remains intact, there are no signs of spillage and inflammatory response. Necrosis is the alternative to apoptotic cell death. They are the complete opposites. In necrosis, the cell swells and the plasma membrane breaks, spilling out all of its contents, which may cause an inflammatory response. Whether a cell undergoes apoptosis or necrosis, it is dependent on the level of stress the cell is experiencing. In humans, developmental apoptosis is the loss of webbing between our toes and fingers in utero to form individual fingers and toes. As you can see here in frogs, the tadpole tail is removed and is absent in the adult frog. The study lasted for a 4-week period, and the purposes included to stop unregulated cell division of undifferentiated human leukemia cells through apoptosis, to learn characteristics of apoptosis and several techniques to define the components in its induction pathway using a human leukemia cell, to determine if the licorice root extract has any components that affect HL60 cell growth by measuring cell viability, to visualize the nuclear morphology of HL60 cells with a fluorescence microscope following treatment with the licorice extract, and to determine if the licorice root contains a polyphenol or other molecules capable of inducing DNA damage in the HL60 model through a Comet assay. The HL60 cells were seeded into a 96 well plate for the viability assay. Seeding is the process of transferring cells from one vessel to another. The t-test values, which are shown here, were determined by comparing the extract-treated cells versus 70% ethanol-treated cells and untreated cells versus 70% ethanol-treated cells. In order to be considered significantly different, the two sets of data had to be less than 0.05, which is represented on the graph by a star. This figure represents the controls of the experiment on the x-axis and the absorbance values on the y-axis. The average values of the data were used to plot the bar graph. As you can see here, the camptothecin and the licorice root were the two sets of data that were considered statistically significant. The cells were seeded for fluorescence microscope assay of apoptosis. This figure displays the cells following a 24-hour treatment with the licorice root extract under the microscope. As you can see, there were no signs of apoptotic cells, just debris. All the cells died after the 24-hour period. Due to all of the cells being dead under a 24-hour treatment, a 12-hour treatment was conducted, which is shown here. But even then, only one cell is seen as a possible cell that underwent apoptosis. The cells that underwent the 12-hour treatment were stained with cyto16 and seen under a fluorescence microscope, which allowed visualization of nuclear morphology following the treatment with the licorice root extract. The Comet assay was conducted to examine DNA damage in the HL60 cells. The head of the comet, which is this green circle here, represents supercoiled, slow-moving, negatively charged DNA, while the comet tail, which is this part, represents a damaged DNA that can migrate faster to the anoid. The amount of DNA damage is proportional to the length of the tail. In the comet assay treated with licorice root extract, it seemed to produce comets with a score of 2. Multiple tails were easily detected by smaller than 3 inches, including this tail, and this tail. They're all smaller than 3 inches. The length of the tail indicates that these cells are damaged, meaning fragments release that migrate from the cell in an electric field which produces the comment tail. To further the study, a shorter treatment plan should be conducted. During the 24 treatment plan, you could see the effect of apoptosis had on the cells by showing signs of debris, but ultimately there were no apoptotic cells present. Also, during the 12-hour treatment under the fluorescent microscope, there weren't really any cells showing any apoptotic markers. The licorice root extract being tested in a shorter period could allow the visualization of apoptosis in the HL60 cells. A different cell model system could also be used as a comparison just to see if the licorice root treatment varies in other cells. A conclusion that can be drawn from the study is that the licorice root maybe kills the HL60 cells off too quickly to be visually seen. The study I read used breast cancer cells, and they showed signs on apoptotic induction.